All right, this is the second review video for our CCP Bio 1220. And after we talked about natural selection, we moved on to population statistics and phylogeny speciation. So we started off with a review of genetic terms like alleles, dominant, recessive, heterozygous, incomplete codominance. If you don't remember those, go back and look at your notes. So that took us into Hardy-Weinberg. The Hardy-Weinberg equation is an equation that is statistical in nature, and it helps us to predict what percentages of population certain alleles will be. And if those alleles match what is actually there, we know that a population has not evolved. If the alleles that are present in the population do not match Hardy-Weinberg, then we know our population has evolved. And in order to not evolve, we talked in the last video about the forces that drive evolution, and we said that you had to have, or the things that would cause it were non-random mating, mutation, natural selection, small population, and movement. So you have to have the opposite of those for for change to not take place. So you cannot have any natural selection. You can't have any mutations that occur. You can't have any movement between populations. You have to have a very, very, very large population, and the mating must always be random. Now, we know that in actuality, this does not happen very often, but so you might think, well, what's the purpose for the equation? What good does it do us? It can show how much a population has evolved, if it's a small amount, if it's a big amount. So there are benefits, and it can give us estimates on the percentages of alleles in a population. So again, for Hardy-Weinberg to be in effect, you could have no evolution, and to have no evolution, you have to have these 5 characteristics at play. The equation itself is p squared plus 2pq is q squared, where p is one allele. Now, often it's the dominant and Q is the recessive. But that's not always the case. If we have codominance, then we don't have a dominant recessive, but P and Q are two alleles. Now, the one equation that always holds true is that the percent of P plus the percent of Q has to equal 1. So we say P plus Q equals 1. It doesn't matter if we're talking about Hardy-Weinberg or looking at an actual population. This is always going to be true. This is only going to be true when we have a population in equilibrium. So P squared is the frequency of the homozygous dominant genotype. This is the allele. So these are alleles. These are genotypes. And you have to understand the difference by the fact that we're talking about how much this allele shows up and how much this allele shows up to our dominant genotype. Here's a heterozygous recessive. Now, if we're doing codominance, these are just we call them our homozygous genotypes. And then the 2pq, we multiply 2 times p times q. That gives us the frequency of heterozygous. Now, it's not going to be on the final, but I want to put it in the review that we looked at problems in the homework where we had, and this is a codominant problem. And the reason we pick codominance is it's easy to see what alleles we have. But here we've got a population, and we know the number of individuals that have each specific genotype. And so we can find P and Q from this data. And using that PQ, then we can calculate our Hardy-Weinberg. And this is a predicted, right? So we get a prediction. And then we can go back and we can look at it with the actual population. If these two match, we know that there is no change, no evolution. Again, I use the evolution rather loosely because we know evolution is usually a longer period of time. But if they don't match, we know a population is changing or evolving. So again, I'm not going to put from this aspect on the final, but it's just good to remember. What I will ask you is to use the Hardy-Weinberg to find and to predict genotype values. So for example, here I have a population and I know my homozygous genotype is 0.366. So remember it is our homozygous dominant plus our heterozygous plus our homozygous recessive. Whoops, it's an A. Where's my eraser? Give me a second here. Whoops, it's not my eraser either. Golly gee. Okay, I'm not having any luck. I know I've got an eraser here somewhere. There we go. We know that this is equal to Q squared. We know that this is equal to P squared, and we know this is equal to 2PQ. That means that this is equal to that. So I know that Q squared equals 0.36. So if I want to know the frequency of A, I have to take the square root of 0.36, and for that I get 0.6. That's my Q value. I know that P plus Q equals 1, so my P value must be 0.4. So if I want to know this is going to be P squared, which is going to be 0.16. And if I want to know my heterozygous, that's going to be 2PQ, which is 0.48. And again, we're going through these quickly as it's just a review video. In this case, we have flowers can be purple or white. So my dominant is capital P, my recessive is lowercase p. And if I have white flowers, that means I have to be PP. And I have 18 out of 42. Sorry, 142 there, which gives me a Q squared value of 0.126. So if I want to know Q, this is Q squared, I have to take the square root of the 0.126, and I get 0.356. P plus Q equals 1. So P plus 0.356 equals 1. That means my P has to be equal to 0.644. And so my heterozygous is going to be 2PQ. That's going to be 2 times 0.126 times oops, I'm sorry. Yeah, let's try that eraser again. Oh, I'm having so much, there we go. 0.356 times 0.644. And that's going to give me 0.458. And then they want you to check your work down here. So you can't expect to see some prediction problems like this. And again, what we're doing here is we're doing predicted. You know, if we had the ability to look at all of the individuals or to do a sampling method of the individuals, we could see if our predictions match the actual. From there, we moved on to speciation. Speciation is the arisal of a new species. And we talked first about the definition of species. We said biological is the most common. It's the ability to successfully mate is the definition. And we said phylogenetic is where we're taking our genetic similarities. Morphological is appearance. The advantage to biological, this is the most common. The advantage to morphological is it's quick. The advantage to genetic is it's accurate. The disadvantage is this is very expensive. It's very time consuming. The biological doesn't work for asexual reproduction or extinct species. And the disadvantage there is that what traits do you use? And is the trait you're using actually valid for what you're analyzing? Also, this works for asexual reproduction, as does the phylogenetic. Biological, you must have gene flow. You must have the ability for gene flow to take place. From there, we talked about how this can occur, and we said there's 3 different forms of divergence. So the most straightforward is allopatric, where you form some kind of geographical barrier and they become isolated. So we form, this is species one, this is species two. When you bring them back together, then you can get some hybridization that takes place in between, but overall they are different species. Parapatric is when you have a new niche that is opens up. So what do we mean by that? So like maybe these are in the next picture we're going to see, let's say we have ground frogs. And let's say that some of the frogs move into the trees. That's a new niche. And over time, because they're separated geographically, the tree frogs actually become their own species. And eventually, when you open back up, there can be some or intermixing. Sympatric is, you're talking about basically here a disruptive selection where you maybe have two extremes. So, for example, if I have a population of bats and one group of bats does a lower frequency mating call and another group does a higher frequency mating call, eventually this group that uses a different frequency will become so enmeshed in that and so immersed in that that the other bats the other bats won't be able to recognize it, and we get this sympatric. We have on the next page again examples. So allopatric, here's our division. Parapatric, here's our tree frogs going in, our frogs that are on the ground going into the trees. And sympatric is we have something that causes a differentiation. Those factors can be as follows: our isolation mechanisms. We can have a temporal, two species made at different times of the year; ecological, different habitats. So this is the one we just looked at with the frogs. So they have different habitats. Behavioral, this would be with the bats. We've got different mating calls. Mechanical, so certain breeds of dogs can't mate, Chihuahua and a Great Dane, or sometimes snails have different shells that go in different directions and they can't physically mate. This is pre-zygotic. This is before fertilization occurs. Post-zygotic is after fertilization occurs. So maybe you produce a hybrid, but the hybrid dies. Maybe the hybrids are sterile. So in the case of, and they give you examples here, you can read back through that. Or maybe over time the hybrids break down where the first generation can reproduce, but then as you go on, over time, things deteriorate and your ability to reproduce diminishes. The last thing we talked about were cladograms. We went over some terms. So the root here is the origin. We've got branches, we've got branch points, we've got sister taxa, etc. One of the things I want to talk about here is the basal taxon versus an outgroup. So an outgroup roots the tree. It's the most distant, it's got the greatest distance, and it's not necessarily related to the taxa that we're looking at. So it's like the closest ancestor to the group that we're analyzing. A basal taxon is the earliest taxa within a group. So it is related. So the root, and I think I have an example on the next page. It might help a little bit. No, I don't. Shoot. So let's say an example here would be if these are all cats, big cats, lions, tigers, leopards, etc. The root might be a wolf. The basal taxon would be the earliest common ancestor. So I'm just kind of going off of a supposition here, but let's say this is a saber-tooth tiger, right? We know those have gone extinct and they're very early. And then you would go into, whatever flow we would have there, maybe a regular tiger, then a lion, et cetera, et cetera. So you need to know the difference between a basal taxon and an outgroup. They're not the same thing. Here's an example. We have bacteria, archaea, and eukarya. And again, we'll deal with these more later. The last thing we talked about was monophyletic, paraphyletic, and polyphyletic. Monophyletic, your group has all of the taxa and a common ancestor. So you see, we come off here, we've got one branch. Here is, I'm coming off of one branch, but a paraphyletic only has part. So if I wanted to be monophyletic, I would have to include this. Polyphyletic, I have this branch, but I also have part. This is branch one, this is branch two. Polyphyletic, I have portions of two different branches. So, and we've got definitions written out down here. But you will see some questions on monophyletic, paraphyletic, and polyphyletic on the exam. And again, one branch and all of its resulting taxa is mono. Part of a branch is para, and multiple branches is poly. And again, here we go, mono, one branch and all of its that come off. B, this one shows it a little better, I think, the para. And then the poly, I have this branch and I have this branch. So I have two different branches and I'm getting multiple groups from two different branches. Here we go. We have another example. I don't think I want to do much here. And that's the end. Okay, so that takes us through the first two homework brackets and the first 9 weeks. We'll continue on with ecology in the next review video.